The joy of learning biology, right here online for you and me. Explore the wonder.
Let's dive into the A-level cycle, a key process in A-level biology. It starts with alpha-oxaloyl acetate.
The Krebs cycle, also known as the citric acid cycle, is a crucial part of cellular respiration, conversion of pyruvate to acetyl coenzyme A. Before entering the Krebs cycle, pyruvate is transported into the mitochondrion by active transport. This step is the link between glycolysis and the Krebs cycle. It begins by decarboxylation. The carboxyl group of pyruvate is removed and released as carbon dioxide. This is the first step where carbon dioxide is produced during respiration. Oxidation. The remaining 2 carbon fragments are oxidized to form acetate. During this reaction, electrons are transferred to oxidized NAD, reducing it to NADH. Formation of acetyl coenzyme A. The acetate is attached to coenzyme AA, forming acetyl-CoA. The bond between acetate and CoA is unstable, making the acetyl group highly reactive and ready to enter the Krebs cycle. Summary of pyruvate conversion. Input: 2 pyruvate molecules from glycolysis. Output: 2 acetyl-CoA, 2 carbon dioxide molecules, and 2 NADH. As we continue, do not forget to subscribe to this channel, like, and share the video. We also have end-of-cycle assessment items. The Krebs cycle, citric acid cycle. Once acetyl-CoA is formed, it enters the Krebs cycle. The cycle consists of a series of enzyme-catalyzed reactions that ultimately release energy and produce high-energy electron carriers, NADH and FADH₂, and ATP formation of citrate. Acetyl-CoA combines with oxaloacetate, a 4-carbon compound, to form citrate, a 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase isomerization. Citrate is converted to its isomer, isocitrate, by the enzyme aconitase. This involves a loss of a water molecule and the addition of the lost water molecule to cause the rearrangement of atoms, oxidation, and decarboxylation. Isocitrate is oxidized, releasing carbon dioxide and forming alpha-ketoglutarate. During this step, oxidized NAD is reduced to NADH. Second oxidation and decarboxylation. Alpha-ketoglutarate is further oxidized, releasing another carbon dioxide, followed by the addition of a coenzyme group forming succinyl-CoA. Again, oxidized NAD is reduced to NADH. Substrate-level phosphorylation. Coenzyme A is displaced by a phosphate group, which is transferred to GDP to form GTP, and then to ADP to form ATP. The remaining compound forms succinate. Further oxidation. Succinate is oxidized to fumarate, and FAD is reduced to FADH₂. Regeneration of oxaloacetate. Fumarate is converted to malate in the presence of a water molecule, which is then oxidized to regenerate oxaloacetate, completing the cycle. During this step, oxidized NAD is reduced to NADH. This is a complete, detailed cycle for revision purposes. Focus on the following. 1. Decarboxylation, substrate-level phosphorylation, formation of NADH and FADH₂. The reduced NAD and FAD carry electrons to the electron transport system for further release of more energy.
This cycle represents the breakdown of a single pyruvate, yet 2 pyruvates are used. Net output of the Krebs cycle per glucose molecule. This yields 3 NADH, 1 FADH₂, 2 ATP, and 2 carbon dioxide molecules. Since 1 glucose molecule produces 2 pyruvate molecules, the Krebs cycle runs twice for each glucose molecule. The net output per glucose molecule is 6 NADH, 2 FADH₂, 2 ATP, 4 carbon dioxide molecules. If you are enjoying this video, subscribe to this channel, like, and share the video. The electron transport chain and chemiosmosis are the final stages of cellular respiration, where most of the ATP is produced. These processes occur in the inner mitochondrial membrane in eukaryotic cells. The electron transport chain uses high-energy electrons from NADH and FADH₂, produced in glycolysis and the citric acid cycle, to create a proton gradient, which drives ATP synthesis through chemiosmosis. It is embedded in the inner mitochondrial membrane. The inner membrane is folded into cristae, increasing its surface area to accommodate thousands of electron transport chain complexes. The electron transport chain consists of 4 protein complexes, 1 to 4, and 2 mobile carriers, ubiquinone and cytochrome C. Mechanism: electron donation. NADH donates electrons to complex 1. FADH₂ donates electrons to complex 2. Electrons from NADH enter at a higher energy level than those from FADH₂, so NADH produces more ATP electron flow. Electrons are passed from one complex to another, moving downhill in terms of energy. The carriers alternate between reduced, when they accept electrons, and oxidize, when they donate electrons. Key carriers include ubiquinone, represented as Q, a small mobile molecule that shuttles electrons between complexes 1 or 2 and 3 cytochromes. Proteins with heme groups that carry electrons between complexes 3 and 4. Final electron acceptor. Electrons are finally passed to oxygen, the most electronegative acceptor. Oxygen combines with electrons and protons to form water. Proton pumping. As electrons move through the electron transport system, energy is released and used to pump protons from the mitochondrial matrix into the inner membrane space. This creates a proton gradient, high proton concentration in the inner membrane space, and low proton concentration in the matrix. Chemiosmosis. ATP synthase is a large protein complex embedded in the inner mitochondrial membrane. It acts like a molecular motor, using the energy from the proton gradient to synthesize ATP.
Mechanism: proton gradient. The electron transport system creates a high concentration of hydrogen in the intermembrane space and a low concentration in the matrix. This gradient stores potential energy similar to water behind a dam. Proton flow. Protons flow back into the matrix through ATP synthase, driven by the concentration gradient. This flow of protons is called the proton motive force. As protons pass through ATP synthase, the enzyme uses their energy to convert ADP and inorganic phosphate into ATP. This process is called oxidative phosphorylation. As we summarize, let us calculate the amount of energy formed. The figure is part of the mitochondrion, showing the inner membrane, where the electron transport system takes place, the outer membrane and the intermembrane space, where protons accumulate. As electrons move along the inner membrane of the mitochondrion, they create ATP energy, which pumps hydrogen ions from the matrix into the intermembrane space. These protons accumulate and pass down back to the matrix via a special protein called ATP synthase. As water flows through the turbines to generate electricity, the same applies to the protons. As they flow back to the matrix via the ATP synthase, energy is generated, used to add inorganic phosphate to ADP to form ATP. Leave a comment as we continue. As we summarize, let us dive into the calculation. Cellular respiration occurs in 3 main stages: glycolysis, citric acid cycle, and the electron transport chain, and oxidative phosphorylation. From substrate-level phosphorylation, 2, ATP is produced directly during glycolysis. 2, ATP is produced directly during the citric acid cycle. A total of 4 ATP molecules from substrate-level phosphorylation. From oxidative phosphorylation, each NADH donates high-energy electrons to the electron transport system, generating about 2.5 ATP. Each FADH₂ donates electrons at a lower point in the electron transport system, yielding about 1.5 ATP. From the complete breakdown of one glucose molecule, 10 NADH yields 25 ATP, 2 FADH₂ yields 3 ATP substrate-level phosphorylation, 4 ATP theoretical ATP yield. In eukaryotes, about 30 to 32 ATP per glucose, due to transport costs of NADH/ADP ATP across mitochondrial membranes. In prokaryotes, up to 38 ATP per glucose, since these transport costs are absent. Thank you for watching. Leave a comment.